I'm Kella, Head of Cell and Gene Therapy here at Team, and we're here today with Katy Newton. Would you like to introduce yourself?
Yes, so I'm currently CSO at MFX, and also I consult on various things in the cell and gene therapy industry, and I've got a background in working in industry in developing T-cell therapies.
Fantastic. And we'll start with what originally drew you to the industry and what's kept you in it?
Science.
Yeah.
Basically just science. So I just knew I wanted to do science. That's what led me to do the degree I did, which was in biochemistry, and then I did my PhD. Didn't intend to go into immunology because I found it very complicated and hard, but somewhat via gene therapy fell into immunology and vaccinology instead. And then when I finished my PhD, I just realised I didn't want to write grants for a living. I just wanted to do the science. I didn't want to do the writing bit. I just wanted to be at the bench and continue to be at the bench as much as I could, and was lucky enough to get a job at a company called Cell Medica, which was one of the first cell therapy companies in the UK at the time, and that was ageing myself 20 years ago now. But yeah, and I just stayed doing what I enjoyed, and there was never really a plan. It was just I stayed doing what I enjoyed while it was interesting, and what really I loved about. I didn't know anything about biotic when I joined, but what I found that I loved about it was the collaborative effort to one combined goal, which was treating that patient, and then the proximity of me to that patient. So the fact that I then managed to, I was actually in the clean room making cells that would be injected the next morning into a patient to hopefully make them better. And that you couldn't get much closer than that to that science being impactful and really making a difference to people. And so that was what really sparked my passion. And although it was hard work and frustrating at times, ultimately I think that's why most of us are in this industry, not because it's easy, but because there's a tangible patient impact at the end of it, and you get to enjoy doing the science, but there's also a deliverable at the end of it, which I think drives us all because it's very important.
So given that 20 years of experience, what would you say has fundamentally changed in that time from then till now in the industry?
Well, I think if you go back that far, it was very much science fiction, I suppose. I mean, it was like the idea was there. There might have been a couple of phase 1 trials that said it could be a good idea, but I'm not sure anyone, was like, well, how do you make money out of this? I mean, how is that even possible? How is that a real thing? Is that going to be anything for patients on a routine basis? How do you get up that paradigm? Is it going to be just for the patients that really have no other options, and this is kind of a one-off last-ditch attempt, or is it actually going to make it mainstream? I always remember one of my PhD supervisors, when I said I was leaving, he wasn't happy, but also he said, does it pass the granny test? Is that ever something that you would actually give to your granny if they needed it? And I kind of have kept that with me, and I was always like, well, we have to make something that I absolutely would give to my granny if she needed it, because that's what we're working towards. And so ensuring the quality and making things safe and everything, that's the number one non-negotiable before you even get to the science. So I think that was it 20 years ago, and the shift to now, which absolutely, and back then we were building the road. There were no regulations that really knew how to treat in any way this new type of, is a cell a drug? Is it a transplant product? Is it what defines the move from being a transplant product like a stem cell to being a drug that is a cell as well? Where is that line? There wasn't a line then. It was like, so we were going back and forward between the HTA and the MHRA, that collaboration was amazing, because we were basically building that road as we were walking along it. It hadn't been paved before, so we were really just, what's next? And so that was fantastic. And now there is very much a road, and people are treading it, routinely, and it's become far more routine, which is amazing to see, right? That growth, that optimism, and that's been driven by the clinical results we've seen. It's not science fiction anymore. These therapies have tangible changes in people's lives, and treat patients and make them better. And that's just light and day from where we've started to where we are now. Absolutely.
And now I'll move on to some more technical questions that we often get into. One of our joint passions of analytics and how that can help solve some of the continuing challenges that we face, particularly commercial viability in the cell and gene space. And so just wanted to ask how you think in-process analytics really can help us move forward with some of those remaining challenges.
So I think in process development, we've always had very much the view that we try as much as we can to have something as close to what it's going to be at large scale, but you need to do lots of things side by side to test all the different combinations of cytokines, of timing, of different medias, of whatever it is that you're trying to get the best product out the other side. And we use analytics at either end. We very much, what does our starting material look like? And then what does our final product look like? And we have always looked at that and try and work out what gets us the best, what starting material, what conditions then get us the best final product. But we don't very often during that process development process look at what happens in between. What's happening in that time? I'm growing my cells, they're expanding, what do the kinetics look like? How quickly are they expanding, at what time points? When do we start seeing different trends, that could predict the outcome? And I think, there's a way of applying what we're doing now to knowing more about it, but equally, once we know more about it, we can then start to apply more adaptive manufacturing techniques to say, if we've seen something at this point, we can go in a different direction and potentially rescue products that we might not otherwise be able to make. And ultimately, if you're thinking purely not about the patient delivery aspect, because obviously we want to be able to treat every single patient, but if you also put your commercial hat on and the commercial challenges that we're facing, the more things that you make that you can't give, that's more wastage in the system, and that starts to build up and not work as commercially as it should. And so being able to manufacture in a more adaptive way, but also being able to manufacture in a way that we learn from those processes and we learn how to make better products as a result of it, because we are collecting that data, and not just at the high at the manufacturing end, so at the large scale manufacturing end, but moving backwards from there. If we know what we're expecting because we've done it 50, 100, 300 times in a small scale process development run, we know what's normal and we know what percentage of things happen in a normal way, and we know, we can start predicting how successful that's going to be when we translate it up to large scale. So without that data collection at small scale, we're not going to get to large scale. And at large scale, yes, collecting that data is very good, but then you're collecting it while you're running, because you're already running your trial. You can't use that data yet to feed back because you haven't treated enough patients yet to know what's good or bad. So learning that from day dot gives you a much more powerful data set to really start relying on when it comes to making process decisions.
Exactly. And do you think, in terms of realising this potential of in-process analytics, I know you've mentioned before the conversations around this are similar to the conversations that were being had on automation and the potential of that 10 years ago. What do you think we can learn from that previous experience and in terms of in realising that potential?
It's interesting because it was definitely, 10 years ago there was an automation special interest group that I was part of at the time, and it was very much like, yes, we know where we want to be, that wonderful, future time when I will press the button and hang my cells up and come back and it's all been released and it's in a bag and I can just give it to my patient. I know where I am now, which is absolutely not that. But then there was this gulf of, well, how does this turn into that? Like, where's the roadmap for that? Because it very much feels all or nothing. And I think we're still in that hinterland with the analytics. It feels like it's, that there is a very big gulf where we aren't collecting anything, and that it will be expensive to be able to implement anything in that space. So why would I, when you haven't shown me why it's useful? But also I can't show you how it's useful unless we've gathered the data. So it's that catch-22 that in the same way was with automation. Yes, clearly there are advantages to it, but how do I build it in a stepwise way that doesn't feel like I've got to invest millions of pounds in building a black box that fully automates something that isn't set in stone yet? And in the same way with the analytics, I don't want to build a black box that has all the analytics that are relevant today if all of those analytics, change because I've treated a bunch of patients over here and I've actually found that my potency test is completely irrelevant, doesn't tell me whether it's going to have a higher chance of working or not in the patient, and actually it's something that I was measuring over here that's actually becomes the important metric, and then I need to introduce something else. So there needs to be a very modular approach to how we can gradually get to the golden hills over here, without it being trying to build it after the horse has bolted almost, because you're now in phase 3, but now you realise that you can't scale because you haven't automated, or you don't want to be in phase 3 and you realise that you don't have any knowledge of your process because you've kind of rushed there without thinking about all of those analytics. And I think importantly it becomes important to capture all of that, but in a going into kind of data infrastructure and that kind of thing, it's becoming more and more clear that we do collect an awful lot of data. Maybe not completely in process, but you're definitely sampling, you're counting, you're phenotyping, but that's not all necessarily being collated and related back to the trials. Like process development and trials can often sit very separately. So you collect it in QC, you collect it in process development, but you might not be bringing that all together to see a much bigger picture. And so it becomes more about that diligence and dedication to collating all of that data from very early on, and being strict with yourself to do it, even though inputting data into Excel spreadsheets is boring.
Yes, absolutely. And so it's what we need is a phased, pragmatic, cost-effective way to gradually implement some of these techniques, starting with the low-hanging fruit and building from there, so that you can build that momentum, see the results, get the buy-in, and we can be
absolutely
more realistically drive towards that blue sky vision.
Yes, and absolutely. And then knowing what actually is important, if you're not measuring it, you can never find out what's important. So you don't necessarily need to be implementing it straight away as part of your QC testing on day one, because good Lord, to try and automate and make that efficient and validate all of those as QC tests would be insanity. But at the same time, do measure it just outside of the QC box, because it might be a future QC assay. We just don't know whether it's important yet.
Exactly. And then by measuring more things earlier, and you touched on this, you then potentially create a new problem, which is having lots of data. And having the data is one thing, but then you need practical ways to analyse and use that data to get value out of it.
Absolutely.
And create aspects that you can use and feed back into the process, which leads me on to the next topic I wanted to talk about, which is the intelligent data analysis tools that are starting to be used more in this field, the AI, the ML, but also the digital twin models that promise to speed up some of that process and give us some of the answers we need in terms of process understanding and control. So I'd like to hear what your perspective is on some of those techniques and their potential.
I mean, I think it is a brave new world across the board, right? And I think anyone who knows me knows that I am a deep Philistine when it comes to all of this tech, and therefore deeply sceptical, actually. Although I know that it's coming, and no, it's not coming, it's here, it's absolutely here. And the utility of it, I think, is likely to blow our minds, but we need to know how to use it and need to know or have the people who know how to double check it. So it comes back to, I think, a kind of cross-functional approach. Already, things like flow cytometry data that I used to be able to play around with on my own, you now need programming skills to be able to do that. And, so I rudimentary programming skills, but I am not a whiz by any stretch of the imagination. So then you need people both with that science, biology, immunology knowledge, but equally with that, that programming ability, that cross-functional ability that they can apply. So we need the data scientists, and so that have that real deep ability to properly know how to organise and delve into and really look at this data, but working with the scientists to ask really dedicated questions. And so, but I also think that maybe we're not late to the party, we're coming as late to the party as anybody else is on this front, but I think even getting everybody on the same page into talking about what we're actually talking about, are we using the right nomenclature in the first place? I did go to a conference where we were talking about this, but I think people were using AI, machine learning, just big data kind of interchangeably. It was, you just collect all the big data and put it into, is it machine learning? Is it AI? Is it the same thing? And those terms were definitely being bandied around without anyone really understanding the distinction between exactly when each of those things would be applicable and in what instance would you use one over another. And so I think the first thing is having the data that you can test it on or train things on, and then having that very much separated from new data that you can test things on. And that is going to be so important. But I think we're just at the tip of the iceberg, right? And we also need the collaboration, because to get big data, everyone's got a piece of that puzzle. And I think if we are to ask some really big fundamental questions, we need the negative data as well as the positive data. So then it's not just published positive clinical trials, it's which trials didn't work. What did those products look like that didn't have a good enough positive signal? Those get buried and we never see them. And that's going to be as fundamentally important to feeding these learning algorithms, rather than just giving them the snapshot of the positive. That becomes so much stronger if you give them the negative as well. So I think, again, it's a community effort and it's a cross-functional effort. You need data scientists, bioinformaticians, biologists, engineers, all of that coming together to generate the right data at the right time and to ask the right questions of it.
Absolutely. Yeah. First we need good data to put into them, and then we need to avoid that potential pitfall of the development happening in silos or using these tools for the sake of it. They are tools in a toolbox, and we need to use the right tool for the right job and ask the right questions and have the good data to back it up. And I think it could really help us along the way.
Yeah, and to know that, I think, is a cross-functional effort from cross-functional people. I wouldn't necessarily know what the right tool to answer the question is, but I can tell you what the question that I want to ask is. So then it's coming together with the people who have the toolbox, but also the people who own the data and understand the data to really ask the right questions in a collaborative way. Because otherwise, again, we're all siloed, and the sum of our parts is definitely bigger, or whatever the phrase is.
I'm with you.
The combination is bigger than the sum of the parts.
Absolutely. All right, I'm going to steer you away from our favourite topic of analytics and enabling technology to solve the manufacturing challenge, and move on to the more global landscape around the industry and how we're seeing much more of the novel cell and gene assets and some of these enabling technologies we've been talking about coming now out of China and Asia more widely. And how do you think the Western biotechs can or should approach that competition that we're seeing now?
I think rather than view it as competition, it's kind of essential that we view it more as collaboration. Because it's a market, right? It is a huge market, and it's a huge market with huge efficiencies and the ability to move fast. And I think in the same way that we just talked about, the adoption of AI, I think if you don't, you get left behind. And I still think, and that's going to be the same with this level of collaboration. I think it becomes a much more global collaboration. I believe stepwise, you have to be stepwise. Let's, prove the concept, small, close to home, just like make it as easy on yourself as you can. Big footprints come with big cost, no matter where those footprints are. So, let's not go too soon, but equally having the conversations, knowing where next to go, what the strategy would be in the different territories, becomes even more important. And I think it's an opportunity rather than a reason that you would compete. And I also think, interestingly, like we all like to think that there is something incredibly special, like that no one else could have ever found about our very specific way of doing something. And yes, we're all built on that kind of level of intellectual property and really want to protect that at all costs. But ultimately, I think there's enough science and reason and building on, the whole of science is building on the work that other people have done. So to a certain extent, I always look at these kind of therapies because they're so have so many aspects to them that they're very much like baking a cake. There's, you can have more eggs, more flour, less this, more, more sugar, bit more of vanilla over here, or a sprinkling of chocolate over there. But ultimately, they're all perfectly good cakes that come out at the end of the day, and they're all, fit the cake-shaped hole that you're trying to fill. And so, fundamentally, if these products work, is a little bit more of one thing or a little bit less than the other going to be the make or break? I'm not sure it is. So rather than being so protectionist and being very worried about my secret sauce being stolen and replicated, someone could, if they really wanted to, someone could find a way of doing something quite similar, quite, quickly, if they really wanted to. So use it as an opportunity to collaborate rather than a fear of the competition.
Yeah, brilliant. I love that. And so now looking more to the future for the industry, what do you think maybe we're not talking about at the moment that will matter most in 5+ years' time?
Where are things going in 5 years' time? I think, it is definitely going to be this, there's a rush to not a rush. We've been talking about it for 10 years, and it does feel finally we're getting to a place where we can really move things forward, and we've got something that works, and now it's a matter of delivering it. And that is one aspect to everything, that the industry is talking about right now. It's like, amazing, we've got these products. We now just need to be able to make more of them so we can deliver them to more patients, and that's brilliant. And so that is one aspect. But I would really encourage us not to forget about development, not only because it's where my heart and soul is at that development end, but don't forget about the science, right? We need a pipeline. So a product's good enough now to be very efficacious in patients, yes. Could there be a better way of making the same thing, or could we make something that was even more efficacious? I think also yes. So yes, while running with what we've got, we also don't want to forget about the fact that we can actually start at keep working on the science and keep working on that development aspect to make sure that with the analytics, with more, just learning more about how our cells grow, how to get the best cells, what are the best cells, how to get more of them and less of the ones we don't want. We can make these products even more efficacious or more commercially viable because we can make more of them quicker or make less of them quicker if we can get more potent cells. Is it quality or is it quantity? So I think all of these questions, and I think more and more the industry is kind of dividing into those two camps. There's like keep developing, keep bringing through that pipeline because we absolutely need it, but also concentrating on real industrialisation of what we've already got that we know is working in patients so we can accelerate patient access, meanwhile keep bringing the other stuff through as well.
And my final question to you, is there anything else you would like to see more of in the industry or you think's important going forward for Cell and Gene?
I mean, I think not only pipeline of products, but I think pipeline of people is really important. And I think that more and more with the collaboration across the different disciplines, I think that cross-training is going to be absolutely critical. So, we've touched on the need for data scientists and biologists and immunologists to all be coming together, but you've got bioinformaticians in there. There's engineers in the mix. And I think that cross training early on and starting to speak the same language, because I definitely think there are, having worked across the piece with all the different people who've come from all of these different backgrounds, there is definitely a language barrier when it comes to exactly what we're talking about, and coming up with a combined language so we all know exactly that we're discussing the same thing at the same time is really important. And so that ability to either bring people from different fields and train them and bring them through in industry that way, or even fundamentally back in universities and having that option for a more cross-functional training than just the science, but all of the tools that you need to be able to do science in this day and age is going to be fundamentally important.
I love that. Pipeline of people as well as pipeline of products. I think that's a lovely place to finish. So thank you so much for your time today. It's been fantastic talking to you as always.
Well, thank you for inviting me. It's been great. Thank you very much.